Okay, so these are really the three areas that we focus on in the moment. I'm going to talk mostly about the first two. We are developing a Hemophilia B program, which is currently in phase 3 during a run-in period in phase 3. So it is a factor IX replacement strategy where we target the liver and deliver a form of factor IX that is hyperactive and highly efficacious. We're also working in the CNS area with a Huntington's disease program, which we hope to have in the IND approved by the end of this year and in the clinic next year. And then we have a number of research stage programs, which I do not really intend to go through in detail today, given the limited time. But we're working in cardiovascular disease with Bristol-Myers Squibb and have a number of additional programs in development, including a Hemophilia A program, which is not a factor VIII replacement. And we'll be discussing our early pipeline later this year in the fourth quarter during an R&D day. So in addition to our programs, one of the areas that uniQure decided to invest in quite substantially a number of years ago is in state-of-the-art manufacturing. And we did this in our Lexington facility, which is also where the majority of the company now is based, using an insect cell baculoviral platform, which for those of you who are familiar with AAV gene therapy manufacturing, insect cell baculovirus is one of the ways to do this. Another very commonly used way is mammalian cells. And there's a number of advantages to the insect cell platform that we identified, and in many ways uniQure began working in this field a long time ago and helped to develop this area for industrial use and overcome some of the initial challenges which some people still have in their mind when they think of this platform that were when the platform was first created at the NIH around stability and other aspects. So we feel we have really created the industrial scale manufacturing using insect cell baculoviral technology, and we've patented that area quite extensively. We have a scalable facility with up to 2 by 2,000 liter production, and we're fully ready for commercial scale up. I can certainly go through that in more detail if anyone is interested. Now, our lead program, AMT-61, we view as a best-in-class product in Hemophilia B. And several of the things, this is using what is known as the patoa-mutant, which is a hyperactive, naturally occurring form of factor IX, which has about 6 to 7-fold increased activity over wild type. And so what we're able to achieve is with lower doses, we can get higher degrees of factor IX activity when we administer it to patients. And before I go into that in more detail, I also want to talk about our proprietary AAV5 capsid. This also we brought in from the NIH a number of years ago and have made some improvements to it. Many people don't always appreciate that the capsid and the reaction of the patient to the capsid has a very big impact on the efficacy of the product as well as who can be administered. And with many products, you may be aware that upwards of 40% of patients are excluded from treatment based on neutralizing antibody status relative to the capsid in use. And this is commonly seen with AAV8 and AAV9 and many others. AAV5 really does not have this problem, and that is enormously beneficial when it comes to being able to treat really the great deal of the patients, probably the majority, if not all patients. And so that really extends the reach of this gene therapy way beyond what other products may offer. We have tested it already in 22 patients, and in addition to the currently dosed patients in AMT-61, that will go up to 25. In 3 clinical studies and multiple indications, including administration to the brain. This is also the capsid we use in our Huntington's program. Very low prevalence of pre-existing neutralizing antibodies and no evidence whatsoever of T-cell responses. So it's an excellent capsid to be used in gene therapy and with significant advantages over alternatives. So back to AMT-61, our phase 3 program is known as HOPE-B, and this is an open-label, single-dose, multi-center trial with approximately 50 patients being treated with our product. And the study objectives, as I'm sure you would imagine, are to look at the efficacy and safety of the product. We begin the study with a run-in period where we measure about 6 months patients' bleeding rates, and then we will dose the patient. We've already enrolled patients in the study and are continuing to do that. Patients have not yet been dosed, but they will be shortly. In addition to that, in parallel, we are also running what is known as what we're calling a dose confirmation study, and I can describe a little bit of the background behind doing this, but what we are doing, and we've already dosed 3 patients in this study. There's no run-in. We're not looking at bleeding rates before dosing. We're just using this to confirm our selected dose in a handful of patients with about 6 to 8 weeks of factor IX activity level. And I don't have enough time to really go through the basis for doing that study, but when that work is completed, which should be available very shortly, then we'll be in a position to dose the patients in our phase 3 trial. This is a Gantt chart, essentially, of the activities associated with this program, most of which have already been completed. And you can see in Q4, as I described, we are completing our dose confirmation work and preparing to dose patients who are being enrolled in the phase 3 study. This is this may be a build as well, right? Okay. Let's see. Nope, that was it. All right. This gives you a sense of how we would measure the results of that dose confirmation study. From 6 to 8 weeks, you will have not your peak factor IX activity levels, but you'll have close to the peak, and you can use that to anticipate how well the product is performing in this early set of patients. So the expectation is that when we get our 6 to 8 week data, it won't be the peak levels of activity, but if it falls into the range of what's anticipated, then that is adequate for us to move forward with the dosing of the phase 3 patients. I would just very briefly touch on the point that we have also an intellectual property that we acquired from the inventor of the Padua mutant, Professor Paolo Simeoni at the University of Padua, who first saw and created this product. And so that has issued patents in the United States and additional patents pending in Europe. And so it gives us a good proprietary position in this field. Okay, then on to the Huntington's program. Huntington's disease, as some of you may know, is a CNS disease that is the result of CAG repeats, an expansion of CAG repeats in exon 1 of the Huntington protein. It's a relatively prevalent disease for a rare disease. This mutant Huntington is toxic, so you have a gain of function that is deleterious that we silence. And we do this with our product that actually non-selectively knocks down all of the Huntington protein, mutant and otherwise. It uses a microRNA, so we deliver the gene that then expresses a microRNA that then engages the RISC complex to be able to silence Huntington protein. It's a one-time injection into the striatum with our AAV5 product using the AAV5 capsid, and that actually then distributes to various sections of the brain where the disease is active, including the cortex and the striatum. We use MRI-guided stereotactic administration, and we've demonstrated proof of concept in multiple small animals, including a transgenic pig model, which is a good approach to demonstrating efficacy in a large model with the human mutant transgenically in the pig. And we leverage our manufacturing platform with this one as well because it uses the same process as we use with AMT61. So this just gives you a sense that we are delivering the gene to where it needs to be, both in the striatum as well as the cortex. And so this gives us good confidence that we are getting to the right regions of the brain, in that case in non-human primates. This summarizes data that we disclosed earlier this year using the transgenic pig model that I mentioned before, where we now see, again, using the approach that we intend to use in the clinic, delivery of the gene to the right areas, as well as knockdown of the mutant Huntington protein in the right regions of the brain, which is very important. And with respect to this program, the next steps are really to submit the IND, as I mentioned before, this year, and begin our clinical trial likely next year. And we have achieved orphan drug designation for this program, along with both in Europe and the United States. And we have completed our toxicology studies, so we're in a good position to get the IND submitted and hopefully approved. And the only other comment I would make is that given we are in a phase 3 trial with our Hemophilia B program, we are also beginning to build out a commercial infrastructure. So that's a bit transformational for the company right now, with the plans to be able to commercialize that product when the time comes. So if there are any questions, I'd be happy to take them. Thank you very much.